Okay, so today we're going to be doing lab number 3, which is metric system. There's a handout that'll be given out in class or posted online. I'll post it online as well as give it out in class. We're going to talk about the metric system, cell structure, and that's Unit 5, Activities 1 and 2 in your book, and then we'll do the rest of cell structure, which will be mostly mitosis. So it's a fairly short one next time. And then we'll talk about the compound microscope. That's Unit 4. And if we're in class, we've usually done what's called a wet mount of cheek cells. And so you scrape the inside of your cheek with a toothpick and then smear it on a microscope slide and look at it under the microscope. We stain it with a little blue dye, and you can actually see your cheek cells. So I don't think we're going to do that because of the virus. So we're probably going to just have to look at pictures of that. I have some pictures I took last year before through the microscope of these. So you can look at we can look at pictures of those as well as the ones in your book. And then we're going to talk about the function of organelles. Okay, so next slide. All right, talking about the metric system. 95% of the world's population uses the metric system as part of the International System of Units, which is abbreviated SI because it's French, is what the abbreviation comes after. So it's Système International des Unités as the official system for measurement, except for the everybody uses it except for us and Liberia and country that used to be called Burma is now called Myanmar. The SI system is based on 7 units: meter for length, gram for how something weighs, Kelvin for heat, ampere for electricity, candela light, second in time, and mole is a number of units, usually of molecules in a, is usually how it's used, molecules in a volume or in a it's actually the number of molecules, and there's not really a unit with it. So it's number of, so a molecule is weighed as the number of grams of it per mole, referring to a number of units of that molecule. So we're not going to use that in this class. You'll use that in chemistry, or if you do biochemistry and start looking at macromolecules. Anyway, the meter. What we're going to talk about is the metric system. So meter, metric system means it's based on the meter. The meter is an agreed upon standard of length from which all the other measurements are derived. The gram and weight and volume are derived. So let's see. A cubic centimeter. So centimeter is a fraction of a meter. So centimeter, centimeter, and centimeter. So a little cube of centimeter, a little centimeter cubed, is equal to a volume. It's a portion of a liter. Liter is volume. So we'll talk about this here in how all these things are derived. But a cubic centimeter is equal to 1 milliliter. So the basic units for the metric system are the meter for length, liter for volume, gram for mass, and Celsius for temperature. The great part about the metric system, it's so much easier to use than the system that we use because it's divided into larger or smaller units, and they're all divisible by 10. So it's all multiples of 10 are divisible by 10. So it makes it really easy. So you convert units within the metric system. All you do is just move the decimal point to the right. Let's see. I don't know, what am I doing on the screen? I'm going. That'd be left for you, and then right. So yeah. So anyway, it's so it makes it easy to convert from the prefix for a unit to the next prefix. So you go from like meter, and you have centimeter or decimeter, centimeter, and millimeter, and those are all divisible by 10. So we're going to talk about that and show how that works here in the next few slides. All right, next slide. So, but start off, so what is a meter? We're talking about the metric system, meter, and everything's based on the meter. What is that? Where did that come from? Well, it was defined in 1790 by the French. they just came up with a definition, and they got together and said, we're going to define it as 1/10,000,000th of the distance from the North Pole to the equator. Of course, running through France, since it's the French are defining this. And it's there's a group of people in France, and they were called the savants, 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 and they were the smart people. So they in France, and they came up with this idea, and they decided this is what it's going to, the length of the meter is going to be. The problem is that how do you know what the distance is from the equator to the North Pole? And there's water in between you and the North Pole, so how do you get across there? And actually, you can't walk it. So you have to walk it, you have to take a boat, and then walk some more, and it's really cold at the North Pole. And this is, the 1700s, and so you're like, we don't have, they didn't have fiber-filled jackets, all that kind of stuff at that point. So anyway, so what they did was they decided to measure a fraction of the distance from the equator to the North Pole, and then use that to define the meter. So what they did was they commissioned a quest. And so it took 7 years, and two guys, Pierre Machin and Jean-Baptiste Joseph Delabre, measured the distance from Dunkirk, France to Barcelona, Spain. Now this happens to be an exact north-south distance, and they measured the distance between these two cities. And so based on this distance in 1799, so it took 7 years for them to do it, and the problem was that there was like revolution, and there was a lot of suspicion. These guys are carrying around these scientific instruments, and they're measuring things. And what they're doing, they go up in church steeples, and they'll use a telescope, and they'll look at a mountain top that's way off some distance away, and then they'll measure that distance, and they'll triangulate. And they actually drew these triangles on sheets of paper, and to use those to measure the distance between point A to point B. Well, you're up in the tower. You've just had the French Revolution. You're up in the tower, and with a telescope and all these other weird instruments, and so people got really concerned about it. So they would confront them, and these guys were thrown in jail and almost beheaded a couple of times. So this is one of the reasons it took so long, because they kept running into trouble. But they were always able to convince people that they weren't actually doing anything wrong. And what made it worse sometimes was that they had a commission from the King of France to do this. However, the king wasn't well liked, and so anyway, long story short, they finished the quest and measured this distance from Dunkirk, France to Barcelona, Spain. And in 1799, the French produced a platinum metal bar, and this was going to be the standard prototype meter. Turns out that when you actually measure all this stuff, it turns out that their definition meter turned out to be 0.2 of a millimeter short. So it's actually not too bad. They did pretty well, considering all the things they had to go through to actually get this measurement. So what's interesting is that not everybody accepted the meter at that point. And I should say the reason you did the meter was because in France there were 10,000 different ways of measuring things. So you went, if you were in Paris and you wanted to buy a measurement of grain, well, they would give you a sack of grain. But if you went out in the country, somebody gave you a sack of grain, but they weren't defined. So you might have a smaller sack in Paris than you got out in the country. And so, and depending on who you went to, everybody defined it differently. So they wanted a standard measurement for the entire country so that they could standardize trade. That's really how why this all came about. And the reason they picked using the Earth as the basis of measurement, they didn't want to just define it because no one would accept that. So what they did was they defined this as a fraction of the Earth's surface, so that it would be a universal measure for everyone, and everyone would accept it, say, oh, okay, this is related to the Earth. So, anyway, eventually people started agreeing to use this measure. And you'll notice that in 1875, 17 countries agreed on the uniform system of measurements and signed the Treaty of the Meter. You'll notice that it's 1875. So this is 76 years after the French produced a standard prototype meter. So if you think we're having like a lot of disagreements now, just realize that disagreements in humans have been going on for a long time. So it took 75 years for 17 countries to agree that we're going to all use this. So we have the Treaty of the Meter, which still stands. So in 1889, they made a meter bar and made it out of platinum and iridium, and there were 17 of these made, and one was given to each of the countries. The official one was the one in France, and the official measurement is defined as the length of the bar at 0 Celsius. And they added iridium because it doesn't shrink quite as much when it gets cold. So anyway, and what was kind of fun about this is that every year or two, people would bring back their meter bars, and they'd all travel to France, to Paris, and they'd all show up there, and they'd all measure their meter bars against the standard prototype that was in France. And so, basically, bring back your bar, you travel all the way there, bring your bar, show that it's the same, and then you have a big party. So they had a big party every year, and this still goes on. They get together and they have a meeting over in Europe somewhere, usually Paris or around there somewhere. So, and it's for the International System of Measurements. So anyway, so in 1960. They got away from using a metal bar, and they decided to, the International System of Measurements decided to define the meter based on wavelength of light. And they picked the Krypton 86 laser excited Krypton 86 is gas excited by a laser, and so the wavelength of light emitted by that was used as the definition of a meter. So you use the actual wavelength of light. So it's a standard measure, because we know from Einstein's theory of relativity that it doesn't matter what direction you're observing light and you're measuring it, the speed of light is always the same. So they defined it based on the wavelength of light in 1983. They defined it using the speed of light. So, and that's a constant no matter what your reference frame is, based on Einstein's theory. So the meter is now defined as 299,792 458th of a second. So that's the distance light travels in meters per second. So anyway, so that's how a meter got defined. It went from measuring across the surface of the Earth. And also, you had to remember the surface of the Earth was also the Earth is not a round sphere; it's a flattened kind of sphere because we're spinning, and so the equator expands out a little bit, and so the North and South Pole are kind of squished together a little bit. So it's kind of a flattened sphere. So you also had to adjust for those measurements for that too. So we've gone from physical measurements to a measurement of a physical property now. So we've done this for all of the other units as well. The last one to be done was the gram, and we actually had the prototype kilogram, and they've redefined that based on basically gravity. So, and that definition went into effect. I think it was last. Was it 2019 in the spring, I think. So anyway, so all of the major measurements are all now based on physical properties. Okay, next slide. So this just shows you the map of where Delambre and Machan both their expeditions went, and so Machan went from Barcelona, and he goes north. Oops. So he goes, there we go. So Machan goes north and Delambre goes south. Okay. And they meet, I'm not sure if I can find it on here. I can't remember. It's, let's see. Machan went one of these guys went a little farther than the other one. I think they met in Rodez down here. I think it's where they met. So anyway. So that was there. You notice all these little triangles. So what you do is you measure from, say, a church steeple here to the top of a mountain, and you find some other landmark over here, and then you have a little triangle. and then you have another triangle, and another triangle, and a triangle, and triangles, all like that. And since, oops, sorry about that. And so since you calculate from the length of the sides of the triangles, you can calculate the distance from here all the way down. I can't draw a straight line, sorry. So you can calculate that distance based on the triangles that you've used, or that you've mapped out using your surveying equipment. Okay, next slide. And that whole thing is actually described in a book, and the book is called The Measure of All Things. That's what it's called. So anyway, so it's an interesting book. It talks about all their adventures and getting thrown in jail and all that kind of stuff. So, all right. So now that you know what the meter is and how it's defined, talk about the metric line. So what we have is this is your basic unit here. Okay, here's a basic unit. So this would be meter, or gram, or liter. Okay, so that's why I put a U right up here. Okay, and so on the right we have small letters. Okay, and these are the prefix to the word. So a D would be deci. Okay, so here's deci. C would be centa. M is milli. And then we're going to skip a couple, and we'll go, so we use a Greek letter, micro, skip a couple, and we get down to nano and angstrom, okay? And you're only going to worry about micrometers, microliters, micrograms if you're doing like molecular biology. I used to use nanograms and micrograms of isolate nanogram, or isolate some micrograms of DNA, but use maybe a few nanograms of DNA to do some experiments. So, anyway, so you can use these prefixes in front of any of the words. So you can have, if your basic unit here is say a meter, then you can have a decimeter, you can have a centimeter, you can have a millimeter. Okay, and those are all abbreviated. Just use the M, so it's millimeter would be mm, centimeter would be cm, and decimeter would be dm. Okay, so deca would be 1/10 of a meter, centimeter would be 1/100th of a meter, and a millimeter is 1/1000th of a meter. So that's why we have this line here. So this is a tenth. Okay, so this is divided by 10. So this is another divide by 10. Okay, 1/10. So this from the unit to here is a 1/100th. Okay, and this would be another divide by 10. So this would be from here to here would be where do I want to put it? 1/1000th. All right. So this is our the way that you look at your units when you go for your meter. When you're doing a meter and you're looking for how many millimeters do I have, how long is something, how many millimeters long is something, you can convert that to centimeters or decimeters by multiplying by 10. Let's see, I'm going to take that line, so I'm going to go this way. It's backward. The camera's backwards from the screen. So let's see. If we go, go that way, you're multiplying. And if you go this way, you're dividing. Okay. So let's see. Then on the other side, we have these units here. This is kilo. This is hecto and deca. Okay. So this would be times 10. Okay, and this is also times 10 from deca to hecto, and this is from hecto to kilo is times 10. So this would be from here, to there is times 100, and from here to here, that's times 1,000. Okay, and I have these things down here. So it's times 1,000 times 100 times 10, and this is divided by 10 divided by 100 divided by 1,000. To get from a meter to a millimeter, you divide by 1,000, or you go from millimeter to meter. That's millimeter to meter. I'll do it for you guys. That's you multiply by 1,000. If you want to go from kilo, kilogram to a gram, then you're going to multiply by 1,000. If you want to go to how many grams in a kilo, then you go you're going to divide by 1,000. So the way to remember these abbreviations up here is you can use this little phrase: King Henry died unusually drinking chocolate milk. So kilo, here we go. So kilo, hecto, deca. Here's your basic unit. U right there. Sorry, U. And deca, centa, and milla on this side. So larger unit to a smaller unit. When you change units, the decimal point moves to the right. Okay, because the number gets bigger. And if you're going from a smaller unit to a larger unit, then the decimal point moves to the left because the number gets smaller. So you have so like 1 meter is equal to 0.1 decameters. 1 meter is equal to 0.01 centimeters. 1 meter is equal to 0.001 millimeters. So, I'm sorry, I did that wrong. 1 meter is equal to let's see, 10. Oh, sorry, I was doing the decameter. Sorry, I was right. Sorry, I'm like, sorry, got myself confused. Sorry about that. 1 meter is 0.1 decameters. Okay, so 1 meter equals 0.01 hectometers, and 1 meter equals 0.001 kilometers or kilometers. That's usually what we say. So same thing going the other direction. If you have 1 meter is equal to 10 decimeters equals 100 centimeters equals 1,000 millimeters. Okay, so if you're going from a smaller unit to a larger unit, you move this point to the right. It just moves in the direction that you're on this number line. So if you're making 1 meter is equal to 10 decimeters, so you move the decimal point to the right. And if you're going from a meter to decameters, then you move the decimal point to the left. All right, so next slide. Here's another metric conversion chart just showing the same kind of thing. So you have your basic unit is here, whether it's meters, liters, or grams. So deca would be 10 of those, hecto would be 100 of those, kilo would be 1,000 of those. And here deca is you'd have 1/10 of those, centa would be 1/100th of those, and milli would be 1/1000th of those. Okay, next slide. Here's another way of looking at it as well. So here's your basic unit, is this dark line right here in the middle. So let's say it's a meter, and so it's 1. So deca is 10 meters. So, or how many meters are there in 1 decameter? There are 10 meters in 1 decameter. There's 100 meters in a hectometer. There's 1,000 meters in a kilometer. And then the other way, how many decimeters? Let's see. How big is a decimeter compared to a meter? It's 1/10 of a meter. A centimeter is 1/100th of a meter, and millimeter is 1/1000th of a meter. And that's mainly the ones that's the only ones we're gonna look at. We're not gonna look at the other sizes in this class. What I want to do is introduce you to the metric system of measurement. And you can use these prefixes for any of the things. It can be meters, grams, or liters. So you can have a milliliter. So that's 1 milliliter is 0.001 liters, or a liter is 1,000 milliliters, or a kilogram is 1,000 grams. Those kind of things. Those are the things that we're trying to get across here. Okay, next slide. Okay, so we'll have a little. Well, some of those conversions will be on the quiz. I have practice questions online that you can take a look at and do. So, okay. So now we're going to go on to the cell. What is a cell? So, I have a little joke here. So that's a picture of someone's test. Supposedly, I pulled this off the internet, so you don't know if it's true or not. So draw a plant cell, identify its most important parts. So a plant cell. Here you are. It's a plant inside a cell. So I know, bad joke. All right. Sorry, I gotta keep myself entertained here. I'm, by myself doing these lectures, it's not really that much fun. So it'll be much more fun in front of you guys. All right, next slide. Okay, so here is a slide or a diagram that's in your book, and I have my glasses on to see this because these letters are so dang small. What I did was I've taken this slide and I've broken it up into little smaller chunks and enlarged it so you could see it. But what we're going to talk about are all the structures within a cell. A cell is the unit, is the basic organizational unit in your body. Okay. So it's and your body's made up of billions of cells, and these cells they each have a function. And you have groups of cells that they group together, and you have a similar function with groups of cells. And so when you group enough cells together, you get an organ, okay? And a level of organization below that is a tissue. So we're going to talk about what cells are. Then we're going to get into tissues, and then into organs. And then we're going to talk about organ systems and talk about how various organs fit together and do a overall function. But we got to start somewhere. So we're going to start with a cell and what's inside a cell, because this will help you understand how these cells function inside, how a collection of cells can work together to do their function. So we have one of the first things you want to know about is there is a wrapping around the cell, and it is called the plasma membrane. Okay, so here's the plasma membrane. A plasma membrane is it's called a lipid bilayer. And the lipid bilayer, lipid is fat, and there's a certain organization to it. It's a bilayer, bi meaning two, so it means two layers. And there is what's called a head and a tail. And then here's a tail and a head. And there's a head and a tail, and a head and a tail, and a head and a tail, head and a tail. And these tails are attached, and a head and a tail. Okay, so this is what it looks like. So you have these heads. This is called a hydrophilic head, and this is called a hydrophobic tail. Okay, so the hydrophobic, hydro meaning water, phobic meaning fearing or hating. So the tails orient towards each other. They flip towards each other, and the heads, imagine my wrists are the heads, they're sticking out towards water. So there's water. There's water out here, and there's water inside here as well. Okay, so this would be, let's make this so let's make this outside the cell, and this be inside the cell. Okay, so you have water on the inside and water on the outside. So these things are going to self-orient, and the hydrophobic tails are going to point towards each other, and the hydrophilic heads are going to stick out towards each other, because hydrophilic means water-loving. Okay, so you have water-hating and water-loving. So hydrophilic is water-loving. So hydrophilic is loving, and phobic is hating. Okay, and this orientation is important because it makes a kind of a skin, in a way, around the cell and keeps everything contained within the cell. And it also prevents things from leaking in or going in and out of the cell very easily. So like, let's see, so water doesn't pass across this membrane very well. It stays outside because the water-hating parts in those tails, they're like, oh no, we don't like you. And so the water can't get in, there's charges that repel. So anyway, so let's see. So anyway, we have the inside of the cell that has liquid in it, has liquid in there, and the liquid is called the cytosol. Okay, cytosol. Let's see. The cytosol is the liquid part, and inside the cell, you'll also hear the other word of, let's see, where's my cursor here? There it is. You'll hear cytoplasm. Okay, so cytoplasm is the liquid plus organelles. Okay, so everything else that we got on the slide are the organelles. So we're going to start on the right side here. So we have a lysosome. Okay, we have a Golgi apparatus. We have a mitochondrion. We have ribosomes here. We have, those are free ribosomes. We have ribosomes that are bound. We have something called the smooth endoplasmic reticulum, the rough endoplasmic reticulum, and the rough, this is called the rough, ER, and this is called the smooth, ER. And then we have a nucleus, okay? And inside the nucleus, we have something called the nucleolus, this part here. Okay, the nucleolus is where there's a type of RNA is synthesized. And then we have nuclear pores on the surface here of the nuclear envelope. So there and there and there, these are all nuclear pores. And nuclear pores are how stuff gets into it and out of the nucleus. And then we have a nuclear envelope, which wraps around the nucleus. And then we have what's called chromatin. Chromatin is DNA, okay, or deoxyribonucleic acid. Make that an E. There we go. That's DNA. Okay. So inside the nucleus, you have a nuclear envelope around it, some pores, some little holes all around the outside. Okay. And then inside that you have chromatin. You have DNA. And chromatin, when you think of DNA, a lot of times you think of the pictures you've seen, and they show you what's called a condensed chromosome. Okay, like this, or you might see it like this. Okay, when it's doubled. So those are chromosomes. You look like, but when it's in your cell, generally it's all unwound, and so it's kind of this loose, stringy stuff that's all in there. So that's loose stuff is all called chromatin, because you want to be able to get access to the strands of DNA to make RNA off of those. So RNA, and then so from DNA you make RNA, from RNA you make protein. And so proteins are what's used inside the cells. And then another type of organelle is called the peroxisome. Oh, I'm sorry. The nucleolus is inside the nucleus, and it's not necessarily like right in the center like this is showing like a core, but it can be around inside the nucleus. It's where a certain type of RNA called ribosomal RNA is synthesized. So you have, it's also called rRNA. So ribosomal RNA. Then we have a couple other things. We have a centrosome. okay, which is composed of 2 centrioles. And those are used in cell division. So you don't really see them, they're not all that useful at any other type. It doesn't appear. So that's a centrosome and centrioles. And we have an endocytic transport vesicle and an exocytic transport vesicle. Endo meaning stuff going into the cell. That's why there's a little arrow going in. And exocytic stuff coming out of the cell. So what happens is you have RNA come out of the nucleus, or you have RNA produced. It travels out and it goes into the lots of times into the rough endoplasmic reticulum, and the ribosomes that are these little dots that are studded all over the surface here. That makes it the rough ER. I'm just dotting all those things. Those are all ribosomes. They translate the RNA into protein. Okay, and then the proteins, raw proteins, they get wrapped up in a little ball and they go up to the Golgi apparatus. So we have RNA come out of here. So messenger RNA goes to here. Oops, RNA. I'm sorry, let me do that again. So it's called messenger RNA. Goes into the rough ER, and then from there you have proteins that have been made, and those go into the Golgi apparatus. And the Golgi apparatus adds things to those proteins. It adds like sugars is most commonly what's added. And then those proteins can be packaged into a little secretory vesicle, which is a little ball containing this protein, and then that goes and fuses with the cell membrane, goes in and a little ball, and it goes up the cell membrane and attaches, and then it slowly releases. It can release its contents out, or it can just fuse with the membrane, and that stuff, the proteins that are embedded in that little vesicle wall could stay there, or it could like spit out the stuff outside. So it depends on what the cell's wanting to do. If it's maybe the cell's making hormones and it needs to spit those out into the bloodstream, or maybe it's making some proteins that need to go on the cell surface, so it creates those, and those stick out of the cell surface. So there's various ways this could actually happen. The mitochondrion, go back to it. This is usually called what's called the powerhouse of the cell. Okay, and it makes what's called ATP, or adenosine triphosphate. And that's used as an energy storage. That's an energy storage molecule, and so it can release energy when needed by the cell. Let's see. And then ribosomes, we talked about those. Those take RNA and they make protein from the RNA template. And smooth endoplasmic reticulum is mostly for production of like lipids and some cholesterol, that kind of stuff. Okay. All right, next slide. So one other part of the cell that I want to talk about is, or a couple other parts I want to talk about. All the rest of the stuff we talked about: nucleus, nucleolus, glycogen granules. Sometimes you have storage of things inside a cell. You can store fat. You can store glucose. Glucose is usually stored as something called glycogen, and you'll find that in muscle cells. Then you have the cytoplasm. We talked about that, cytosol, plus the organelles, except for the nucleus, rough ER, ribosomes, Golgi complex. Let's see, plasma membrane is there, is here. That's what that little thing is. Microtubules, and we'll talk about those here in a second. Mitochondrion is these kind of kidney-shaped things here. Here's one. Here's another one here. Okay. And we have peroxisomes, and we have lysosomes probably around here somewhere. There's lysosome. Those are two things that are, they break down. The cell can take in things, and it goes into a lysosome or a peroxisome, and those two organelles can break down things that the cell has brought into it, or it can break down things that are inside the cell. Let's say you have a protein that's become damaged, it's not good anymore, so a lysosome can take it and break it down back into amino acids so that your cell can reuse that. And lysosomes can also go and become secretory vesicles and spit stuff back out of the contents of it. Once it's done, they can actually spit stuff out. So, but usually the secretory vesicles come from like the Golgi apparatus or something like that. So, Smooth ER, we talked about that. Plasma membrane there. Let's see what else. Centrosome, we talked about that. And then we'll have, so one of the things we have in the cell, we have something called the cytoskeleton. Okay, so you notice that cells, they're not just this blob, they actually have shapes inside the body. You'll have, some will be cubic, there'll be cubes, some will be like elongated or like rectangular, and sometimes they're roundish or different shapes like that. So, but inside the cell, there's actually proteins that create the structure, and they make the cells a little more rigid inside. And the proteins that do that are called microtubules, microfilaments, and intermediate filaments, and they all produce different shapes, and they can also produce motion as well. So you have a flagellum. There's only one cell in the human body that has a flagellum, that's the sperm, but you have microtubules that cause rotation of the and whip-like motion of the tail, so that the sperm can move up towards the egg. You also have cilia, singular cilium, plural cilia, and so those will move things past the cell, and you'll have those like in the trachea. So you'll have, so you breathe in, you get some dirt in your trachea, and you have a little cilia, and the cells lining the trachea make some mucus, and so the cilia move, they move back and forth, and they move that dirt back up out of your trachea so that you can cough it out. Let's see what else. Oh, microvilli. Microvilli are a special thing. What they do is they increase the surface area of the cell. So let's say your surface is like this, okay? So you have this as your surface area. But if I have these projections on here, now I've got surface area all across here, so I can have a greater absorptive area. I can absorb nutrients across that. So you'll have this, say, in the intestines when you have food passing, so that you have lots of area all through here that can absorb stuff and go into the cell. Okay? All right, so those are your various parts of a cell. Now, a good thing to do is take your lab book and take a cell and draw all the parts on the cell and just draw them in there, and so that will help you a ton memorizing these things. If you don't know these things, if you had basic biology, you may know these things already, but if you don't, it'd be a really good idea to do those. And one other thing to point out is the centrosome, the centrioles, you'll notice, are in kind of this sort of a T shape here. Okay, so that's a centrosome, is when both those pieces are together. All right. All right. Next slide. So here's just a higher magnification. It's not higher magnification. It's I took it and enlarged. Sorry, the diagram that was in your book. So just to show you these, this will be in your online on the PDF. I'm not going to go through this all again because we already went through this stuff. You can follow along with your book in the previous slide when we're talking about those things. Okay, next slide. So this is the top right quarter. Next slide. This is the bottom right quarter, seeing the mitochondria and ribosomes free and bound. So some ribosomes are floating around in the cell. Others are bound to the rough endoplasmic reticulum. And let's see. Next slide. Okay, so it's the bottom left quarter. So we have the nuclear pores, nuclear envelope, chromatin, rough ER, smooth ER, that stuff. So, all right, next slide. So, okay, so this is a little cartoon. The organelles talking to each other. So the nucleus thinks that everybody has to do what he says. Mitochondria are like, no, we don't have to, because actually mitochondria are kind of interesting because they have their own DNA. Because mitochondria are thought to be basically maybe bacteria that were engulfed by early cells way back when, and they survived inside cells, and then and what bacteria have is in their cell membranes, they actually have the energy-producing molecules to make ATP. So it was really helpful for cells to engulf these bacteria, and these bacteria just like are instead of being parasitic, it's now a symbiotic relationship essentially. So, and mitochondria actually have a little piece of little DNA molecule in them, so they can actually replicate themselves. So the mitochondria don't have to do what the nucleus says; they can do whatever they want, pretty much. And then the endoplasmic reticulum is like, oh, I've got so much work to do, because it does. It does a whole ton of work. And the Golgi apparatus is like, man, I'm making all this stuff and I'm blebbing off these little, it's called blebbing actually, yeah. Bleb, you bleb off, or blebbing. You're blebbing off these vesicles off the Golgi, and those go to the cell surface. You also have vacuoles. Vacuoles, they can be filled with things like fat or whatever. So that's why the vacuole is filled. It's filled up with stuff. A lysosome, it destroys everything. Okay, whatever goes into the lysosome, it has digestive enzymes and a slightly acidic environment that goes and destroys whatever in it. Okay. Anyway, so blebbing is usually it's about the surface of the cell, and you have little vesicles coming off and blebbing off like that. Okay, all right, next slide. Okay, so we're going to talk about the functions of the major organelles. We talked about where they are in the cell. So the ribosome, that is we'll look at that structure here in a second. I'm going to look at these in a little more detail. This is just an overview table. This is a table out of your guys' book. So the ribosome, it synthesizes proteins. The rough endoplasmic reticulum, or sometimes abbreviated RER, synthesizes and transport proteins to various places in the cell. It might transport them to another organelle or to the cell surface. Let's see, a smooth endoplasmic reticulum, or abbreviated SER. It synthesizes lipids, steroids like cholesterol, also detoxifies drugs. So you'll find it doing that, like in the liver. Is one place that drug detoxification takes place. The Golgi apparatus, it packages and modifies protein. So you have protein, and you add things to it, and you also maybe package it with other components for transport to the cell membrane. And sometimes you put carbohydrates on the surface of on the outside of a protein, and then you transport that to the cell membrane. So now you have a protein with a carbohydrate sticking off of it. And so we'll take a look at that here in a second, what that looks like. The mitochondrion, it's usually in basic biology, I always refer to that as the powerhouse of the cell. Synthesizes adenosine triphosphate, ATP, and is the abbreviation for adenosine triphosphate. And that's the power source of the cell. So it has a 3-phosphate triphosphate, so 3 phosphates, and the third phosphate is lost fairly easily, and so that can be put back on fairly easily. So you have this phosphate on here, so it falls off, and energy is released when that comes off. So the ATP is used as an energy storage device, and ATP is produced by the mitochondrion. Lysosomes. They are they're called vesicles, which is a little ball, and it has enzymes in there, and those digest proteins within the cell, worn out organelles, or substances that have entered the cell, say like a bacteria or a virus, that can be taken into lysosomes and broken down. Peroxisomes are often used to detoxify toxic substances. They produce peroxide or peroxides that are used to detoxify these things. One of those is like hydrogen peroxide is a simple one, H2O2, and it structurally looks something like this. And so if you've ever had hydrogen peroxide put onto a cut, you have an enzyme in there called it's a type of peroxidase. It's called catalase, and catalase breaks down hydrogen peroxide into water plus oxygen. So catalase is an enzyme, and you know it's an enzyme because it has ASE on the end. So if you've ever poured hydrogen peroxide on a cut, you'll see little tiny bubbles, and those little bubbles are oxygen bubbles. Anyway, let's see. Okay, centrioles. Centrioles, they organize the mitotic spindle during cell division. And centrioles, there's 2 of them. They kind of make a T kind of shape, and so the 2 of them together makes a centrosome. And so that's what that word there means. And we'll take a look at all these things, the structures of these here in a second. The centriole, what they do is the mitotic spindle is a bunch of fibers that come in and they attach to the chromosomes. And chromosomes all line up on the middle of the cell, and then they get drawn by the centromere on each part of a chromosome. Okay, so there's a centromere, and there would be a centromere, so they get drawn and drawn to the edges of the cell. And they get drawn along microtubules. So those are components of the centrioles, and they also form the spindle apparatus during mitosis. And so next class we're going to talk about mitosis and how that works. So the cytoskeleton is made up of microtubules, intermediate filaments, and microfilaments. And all these different filaments, they make a structure, they give the cell the structure that it has. So it may be squarish, it may be rectangular shaped, but what you need to know, I don't need for you to know all these different names and what all these things do. What I want you to know is just that there are proteins within the cell that give it structure, that make that, that give that cell the particular shape that it has. So it's not just randomly this forming in the shape. There's actually things that make the structure of the cell. And these are, and so these proteins are all components of what's called the cytoskeleton. Okay. Next slide. Sorry, I lost my mouse there for a second. Next slide. So the cell or plasma membrane, one thing I want to point out, but it's a flexible, but it's also a sturdy barrier. It surrounds the cytoplasm of the cell. There's something called the fluid mosaic model, okay? So we talked about the lipid bilayer. So you have hydrophilic heads, hydrophobic tails, and then you have the other layer, hydrophobic tail, hydrophilic head, hydrophobic tail, hydrophilic head, hydrophobic tail, hydrophilic head, hydrophobic tail, hydrophilic head. So here's your lipid bilayer. And then you can have proteins embedded in this lipid bilayer, okay? And it can be on one side, it can be on the inside, okay, like that, or it can go all the way across. All right, make that circle better. Okay, so you can have if it goes all the way across, it's called transmembrane protein. If it goes part way, and it's also called integral membrane protein. But what you have these proteins that are in this lipid bilayer, and they can move around. Okay, so that's why it's a fluid. It's a mosaic because you have proteins and lipid together, but it's fluid because these proteins can move around in this lipid bilayer. Okay, and these proteins don't have to be just protein. They could have a little, sugar molecule sticking off the top of it. This guy could have it too. Okay, that kind of thing. And these proteins could be transport proteins. They could be transporting things outside the cell into the cell, or inside the cell out of the cell. So anyway, but I just want you to know about the fluid mosaic model. Lipids, you have a lipid bilayer with proteins embedded throughout, okay? The membrane contains lipid and protein. Lipid is a barrier to entry or exit of polar substances. The hydrophobic tails do not like polar substances. A polar substance that you're very familiar with is water. Water is polar because, here's what it looks like. It's HOH. And water kind of drags electrons a little closer to it, so it has a slightly negative charge, and the hydrogen have a slightly positive charge. So that's what polar means. It means there's a polarity to it. There is a negative and a positive pole, kind of like a battery has in kind of a general kind of sense. Okay, so if you have a neutral molecule, it can flow across the membrane pretty easily. But if you have a charged molecule, those do not flow across the membrane. So you have to have a channel or a transport protein for those. And so you have the integral proteins. They regulate what comes in and out of the cell. So you have integral proteins that can regulate water coming in and out of the cell, calcium ions, potassium ions, all those kind of things. We'll talk about those later on in the course. All right, next slide. So here's what the lipid bilayer looks like. So here's a 3D version of it. So here's protein sticking out. You might have a skinny protein over here, or you might have a fat protein. And you notice there's this hole down the middle. Okay, so that tells you that stuff could potentially go into or out of the cell, or both. It could go flow back and forth, depends on how it is. Okay, here's a fat protein over here that doesn't have any pore in it, any central canal in it, but it does have these balls, and that's some sort of carbohydrate up there, so some sort of sugar. This one has sugars, this one has sugars, these have sugars here and here, okay? And so proteins can be different shapes, but these proteins can move around the cell, so they can actually, if you wanted to, you could have them all concentrate on one side of the cell, if the cell wanted to do that for some reason. Or they might be equally distributed around the cell. And so why would you want to concentrate proteins on one side of the cell? Well, let's say you have a whole bunch of cells that are next to each other. I'm going to draw rectangular cells, and another one here, and another one here, and another one here. Well, and let's say that these cells are, let's say this is the lumen of the intestine here. And let's say this is let's say there's a blood vessel down here. Okay, so this is a blood vessel. Well, I would want all my proteins for transporting food to be up here on the top, right? And I'd want proteins along the side that help hold these cells together. Okay, so I'm going to concentrate some proteins on the sides to hold all these cells together in their little soldier row, lining the whole small intestine. And then I have proteins that are pointing towards the inside of the small intestine. Okay, so I want proteins pointing. This is a small intestine. I want proteins pointing in that are going to take in food and take food into the cell, or take food in. Let's see. I want to turn this into the cell. Okay. All right. So next slide. Here's a diagram out of your guys' book. I thought that the reason I showed the other one, I think, is a little simpler now. So here's your diagram out of your guys' book. So it's showing a part of the cell membrane here. So outside is the extracellular fluid. So that's outside the cell. So this is outside, or it is extracellular, and this is inside, or also called intracellular. Okay, so here's a phospholipid molecule, all right there, right there. So here's your hydrophilic head, and there is your hydrophobic tail. Okay, and then you have what are called all these proteins I show here, except for this one. Well, all these proteins are integral membrane proteins. All of them are transmembrane, except for this guy over here. This guy doesn't go all the way across the membrane. And then you have, so this is a transmembrane, this is transmembrane, this is transmembrane, this is transmembrane, this is transmembrane here, and this is transmembrane here. Okay, they're all transmembrane proteins, but they're sorry, there was a squirrel outside my window making a whole bunch of noise. Anyway, and then you can also, it doesn't have to be just one protein. You could have an associated protein. So we have a peripheral protein. So you have another protein here associated with this protein. All right, so you have this protein here, and you have another one that's on the inside, or you have a protein here and you have another one sitting on the top side of it. So it depends on what functions it has. So you have peripheral proteins related, associated with integral proteins. And then some of these proteins have a pore in the middle like that. So, but you notice it's closed off on the top and the bottom. Okay, so it's not open. So what happens, like one example is like insulin. Insulin comes along and causes that to open up. Okay, and glucose then will now fit inside there, and you'll put glucose in your cell. Open up, take glucose, put glucose in the cell. Open up, take glucose, put glucose in the cell. If that protein becomes resistant to insulin, it now won't open up. So glucose stays out in the bloodstream, and you have high glucose levels. Okay, so you have these proteins. They can be open all the time. They can be like one protein called an aquaporin, where water can be in and out of the cell, or you have proteins that only open up under a certain stimulus, like say for insulin. So if you eat and then insulin levels go up, and so it causes your glucose receptors to open up and start taking glucose into the cell. Okay, so there's some examples of how this all works. All right, so in the inside of the cell, you have the cytoplasm. Okay, all right, next slide. So the cytoplasm is everything between the membrane and the nucleus. Right, so it does not include the nucleus, but it's everything inside the membrane except the nucleus. And it's composed of the cytosol and the organelles. Cytosol is the fluid inside. It's 75 to 90% water. Also has large and small organic molecules, inclusions. You can also have which are aggregates of a material, say like glycogen. You can have glucose that's stuck together in chains, and it's called glycogen. Let's see. And then fats usually don't do as inclusions. They usually are stored. Their fats are usually stored inside inside a storage structure. Let's see. Organelles are any of those subcellular structures that we talked about that have specific functions. The Golgi apparatus, the rough ER, the smooth ER, mitochondria, lysosomes, peroxisomes. Let's see, and centrosome, ribosomes. That's the majority of them. Okay, anyway, next slide. Okay, so just show you what the cytoplasm is. Cytoplasm is between here and there. Okay, so that's the cytoplasm. Next slide. Nucleus, control center cell, contains the DNA to make structural and functional proteins. It has the whole blueprint for you, and every cell in your body has the same DNA except for your matured eggs or sperm, depending on whether you're male or female or male, respectively. And then let's see. So we talk about this: the membrane, nuclear envelope, the nuclear membrane. Two words mean the same thing. It has pores in it, so you have these perforations, these openings in there, so stuff can get out. Mostly like ribosomal RNA and messenger RNA. Nucleolus has it's where you have ribosomal RNA is assembled there, so you have ribosomes. Ribosomes are a mixture of ribosomal RNA and protein together. Okay, so that's what a ribosome is. So the ribosomal RNA is made in the nucleolus, and then it's assembled, and so with proteins that are made down there, and then the ribosomes are then brought out. Next slide. Mitochondrion has an interesting structure to it. You have an outside, and so we saw like the sort of kidney bean shaped structure. Then we have this structure on the inside, like this. Okay, and so each of these projections going in, these are called cristae. Okay, what it does, increases surface area, and you have, so a cristae is, let's see if I can, my hand right, there we go. So a cristae going up and over, okay, and you can have more surface area for, because this is where all the energy production occurs, is along these projections, the cristae, all up and down all the way through. It just gives you more surface area in there, so you can make more ATP than if you just had a smooth ball-like structure. So if you make it convoluted in there, then you have a lot more surface area inside. So, and the inner part is called the matrix. These are called cristae. You have the outer membrane and the inner membrane. The inner membrane is then folded into these cristae. So it's where ATP is produced. Now, I'm describing all this stuff. You don't have to know all these pieces. I just want you to know that ATP is what's produced out of the mitochondrion. Okay, next slide. Sorry. So the mitochondria has this double membrane structure. The inner membrane is where ATP is made. Okay. All right. Next slide. Okay. So what's the big deal about making mitochondria or mitochondria making ATP? Well, you know that every day you are doing two things almost every day, unless you decide not to eat. So you eat and you breathe. Okay. So what is breathing? Breathing is you breathe in air and you breathe out air. Okay. When you breathe in air, air has 21% oxygen. When you breathe it out, the amount of oxygen has gone down a little bit, and the amount of CO2 has gone up quite a bit. So the air you breathe is almost all nitrogen. It's like 78%, 79, almost 79% nitrogen. 70, high 78% nitrogen. There's trace gases of other things. Carbon dioxide, one of those. Then when you breathe out carbon dioxide, you're breathing out a percentage of what you breathe out is carbon dioxide. It's still a lot of oxygen you're breathing out. Anyway, you have this exchange of oxygen and carbon dioxide going in and out of your body because your body is using oxygen and creating carbon dioxide, and that production of carbon dioxide happens in the mitochondria. So then you have your bloodstream to carry the carbon dioxide back to the lungs and expel it when you breathe out, like that. You also breathe out. So you have breathe in oxygen, breathe out carbon dioxide. You also breathe out water vapor. Okay, so every time you breathe out, there's water vapor coming out because you're producing water from the breakdown of glucose. Okay, so glucose is C6H12O6. So you'll notice there's C's. That'll be, and so you make carbon dioxide from them, and O's. You're going to make CO2 from that, and you have H's, so you can make H2O as well. So those are some of the byproducts of breakdown of glucose. Okay, so you eat glucose, and then your cell breaks it down, and you create ATP, and then you expel the leftover products, which will be part of it will be broken down into CO2 and water. Other parts you might have some lactic acid and those kind of things as well that are produced, pyruvic acid, those things. And anyway, so what you get out of each molecule of glucose, you get 36 molecules of ATP. So it's a pretty good energy generation system. Next slide. So here's what happens inside the mitochondria. And I'm going really deep into this. You don't have to know this for the test, but I think it's important for you to understand there's a lot of stuff going on inside the cell. Okay, so you have glucose going in the cell, and it goes in. So the cell takes up glucose and goes through something called glycolysis, and you create what's called pyruvate. Okay, a 3-carbon sugar. So you break a 6-carbon sugar. The glucose is 6 carbons, and you break it down into 2 3-carbon sugars. There's 2 of those. 2, 3, 2. Maybe I should make that a comma, huh? 2 3-carbon sugars. Okay, and then so in the absence of oxygen, okay. So let's say you're running really, really, really hard, and you're going along. You're not bringing in enough oxygen to break down the glucose, so you're going into a anaerobic state. So you have aerobic, where you have plenty of oxygen to break down glucose, or more anaerobic, meaning without aerobic or without oxygen over here. And you get into what's called fermentation. So you break down that pyruvate into lactate, and so the buildup of lactate or lactic acid in your body, and you can get rid of that fairly quickly. It's not too big a deal, but you don't want to stay anaerobic for very long because you don't make as many ATP molecules out of that. You make some intermediate molecules, NADH, NAD+, which goes on, which can be used to produce energy. If oxygen is present over here, pyruvate goes in to the mitochondrion, and you start making NAD and NADH, but you make a lot of ATPs out of all of this. Okay, and you go into something called the TCA cycle, the tricarboxylic acid cycle, and you make more ATPs out of that. So that's how you end up with the large number of ATPs out of a molecule of glucose. Next slide. So you don't have to know this, but if you go on into biochemistry, you'll actually memorize this, and you'll memorize the structures of all these. This is the Krebs cycle, or the tricarboxylic acid, or TCA cycle. And so you don't have to memorize that. You don't have to know this. I just want you to know, here's pyruvate coming in up here, and it goes in, and then you have this cycle, and it circles around. Okay? So, and you're throwing off ATPs through the Krebs cycle. Okay? So here's one over here. And then you have a related molecule called GTP down here. So, all right, next slide. But you don't have to know this. I just want to point out that what I'm going to try to point out during this whole class is that we're going to scrape the surface of this, and there's a whole lot deeper levels you can go in every single topic that we're going to cover. Okay, so don't think that we're going to go completely in depth into every single topic. There's lots more to know about every single thing we're going to talk about. So you don't have to know this for the test. Don't get worried. Just know that there's if you go on and you study this stuff further, if you study biochemistry, if you study anatomy further, or physiology further, you'll need to know these kind of things. Okay, next slide. All right, go on to a ribosome. A ribosome is made of RNA and proteins. I said that a little bit earlier. Fixed ribosomes are bound to the rough ER, and free ribosomes are scattered throughout the cytoplasm. And all those can make proteins. Free ribosomes are out in the cytoplasm so that you can go and make a protein out in the cytoplasm without having it to be packaged and go on a Golgi apparatus, all that stuff. So ribosome is where protein is synthesized. Okay, and you have two subunits of a ribosome. You have a small subunit and a large subunit. So these two, you have a big piece and a smaller piece that fits together. And the RNA appears to go through this whole thing. The messenger RNA will go through here. So this will be messenger RNA. Come out. This is messenger RNA, which is a single strand of nucleic acid. And as it goes through here, the ribosome brings in, I'm just going to draw them as balls, brings in amino acids, brings in individual amino acids that are hanging out here, and it connects them, and based on the messenger RNA, what message it has, it selects different amino acids and puts them together in order based on the sequence that's in the messenger RNA. So that's how you get a protein sequence, or a sequence of amino acids in a protein. Okay, next slide. Lysosome. Talk about this. This is where a vesicle that contains digestive enzymes that break down substances in the cell, such as foreign particles or worn out organelles. So you can have a vesicle that contains fluids, so it brings something in, a solid or a fluid, and then it goes and it breaks it down and it releases whatever's left. So let's say it was a pathogen that was brought in and it broken down by the lysosome enzymes, and then it releases it back out because I don't want that stuff inside me, and it's waste products. So then your body can get rid of it. There's other things that can happen. You can make, from the Golgi apparatus, you can make these vesicles that come up, and it can go one or two ways. One, it can be used inside the cell, or two, it can be sent, it could be sent on out to the outside of the cell, or it could be sent to a lysosome and broken down. Let's say it's nothing that's so let's say a lysosome doesn't have anything in it to break down these cells. So the Golgi apparatus has made some enzymes that break down, that fuses with the lysosome here to be able to break stuff down. Okay, so what I want to point out is just there's a lot of complicated things that go on in here. So we're just, all we got to do is know that there are organelles and they have all these different functions. Okay, all right, next slide. Peroxisome, another vesicle containing degradative enzymes, and you can neutralize toxic compounds. You have enzymes called peroxidases. One of those is called catalase. and it breaks down hydrogen peroxide. And there's actually a question about whether it neutralizes hydrogen peroxide or it produces hydrogen peroxide or peroxides to break down things inside the peroxisome. So, but hydrogen peroxide gets broken down into water plus oxygen. So, if you ever had a cut, people don't do this near as much anymore because people don't pour hydrogen peroxide on cuts all that often anymore. But if you ever done that, had a cut on your finger, say you cut it with a knife or something like that, and so mom poured some hydrogen peroxide on there, you'll see little bubbles form. Those bubbles are oxygen bubbles. Okay, so because the catalase that's in your body, in your red blood cells, and that's in your tissues there, breaks down that hydrogen peroxide into water and oxygen. So that you pour it on there because that hydrogen peroxide is also very reactive, and it'll react and kill bacteria that are on here. But your body also tries to protect your body from the hydrogen peroxide, so it also breaks it down. So your body breaks down most of it into water and oxygen. So anyway, and that's what peroxidases do. Catalase is a type of peroxidase. Next slide. Smooth ER. These membranous sacs that are called cisternae, and they're tubular. Okay, so this is smooth ER, SER. No ribosomes on it. Synthesizes lipids and cholesterol. It can also detoxify harmful substances. Next slide. Rough ER has tubular cisternae as a but these are more layered rather than tubular generally, and you have ribosomes attached, all those little dots that are attached. Okay, so that's site of protein synthesis. So it makes and modifies newly synthesized proteins for use in the membrane or for use in the plasma membrane or for export outside the cell. Next slide. Golgi complex. It stores, alters, and packages proteins for export or use inside the cell. Okay, I'm not going to go through the picture here. You can read that if you want in the PDF. Next slide. Cytoskeleton, we talked about microfilaments, intermediate filaments, microtubules. So those just add structure to the cell in various ways. So I'm not going to talk about them in any more detail. Next slide. Centrosome, we talked about earlier with that, I have a typo on here, containing, put a G on there. Centrioles, so it's a formation site for the mitotic spindle where you drag the chromosome, chromosomes apart. Okay, and then they're two centrioles, 90 degrees to each other. Okay, so it makes it kind of a T. So there you can see that. So you have this way and this way. Okay, all right. Next slide. Okay, cilia, hair-like projections, they propel stuff past the cell. Talk about that in the trachea, moving dirt up your trachea so you can cough it out. Next slide. Flagella, just a type of, it's sort of like a cilia in a way, but it's a lot longer, and it's a single projection off the cell. Okay, next slide. Okay, membrane transport. You're going to go over this in your class and lecture. But I thought I'd do this also in here because it's in your book. So we're gonna do this. Okay, we have there's different types of transport, and we're gonna talk about simple diffusion, facilitated diffusion. There's two types of it, and there's osmosis of water. Okay, so these are all passive. Okay. And this one is active. Okay, so active transport there, and passive transport is all of these. All right, so active transport requires ATP. All right, so here's your ATP molecule right here, giving off energy, and it causes this, in this case, what they're showing is the sodium-potassium pump. So this is important with nerve cells, and so you're pumping sodium ions out and pumping calcium ions in. Boom, 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 boom. You're pumping. So in order to make this happen, you require ATP to do this. Okay, so let's see. The other ones are passive transport. So simple diffusion here, nonpolar molecules, so things that are like not water. So cholesterol can just go right across the cell membrane. Okay, other lipid type molecules can go across the cell membrane. Facilitated diffusion, you can have a couple types of that. You can have ions go through just an open channel. So you have ions, positive ions come back in, negative ions go out and back and forth. A facilitated diffusion, you actually have an ion or and a carrier protein. When the ion binds to it, it causes the protein to change shape, and so it kind of like will spit an ion out or spit an ion into the cell, one or the other. The other type is it's not diffusion, but it's sort of like it allows diffusion to occur, and these molecules are called aquaporins. And these are protein molecules. Okay, here and there's water, and here's a water molecule. Okay, and so this is a hole in the cell that allows water to go into the cell or out of the cell if the cell has too much water in it. Okay, so there you go. So you have passive transport on. I don't know how to do it. Passive transport on the left side, and active transport is there's only one piece where it shows the sodium potassium pump over there on the right side. Okay, next slide. Cell specialization. So we talked about structure and function in anatomy. Structure and function are very closely aligned all the way through anatomy, from the cellular level all the way up. So you have differences in the functions between each cell type. They're reflected by the differences in structure. So here's an example. Skeletal muscle is over here. Okay, and you can see these stripes in here. You can see stripes. Okay, those are called striations. All right, so what you visually see, you have these stripes going across the, all the way down the muscle, and those are actually proteins. They're actually they're there and they've stuck together, and so where they come together, it's a little bit darker. So there's a little stripe, there's a band there. Okay, so the proteins aren't like this; they're actually like this, and so when they come together, it makes little bands, and those pull against each other. Okay, and when they contract, these actually they don't pull, they slide against each other to contract the muscle. Okay, and let's see, here's another example. You have pseudostratified ciliated columnar epithelial cells. These are found in trachea and upper respiratory tract, and up here is the cilia. That part right up there, right next to the black part up here, there. And there, those are the cilia, and so those are in the trachea. So those are the little hairs that are moving stuff past the cells, and so you can cough it out. And each of these round balls, that's a nucleus, that's a nucleus, that's a nucleus, that's a nucleus of a cell. So this would be so there's a cell, and there's different cells in here. We'll talk about this when we get into tissues. We'll talk about what this looks like. So those are all different cells. And then we have, I move me out of the way from me. So this is called simple columnar epithelial cells over here. Simple columnar epithelial cells. These line the digestive tract, and this part up here, right there. Well, let's see, I should point it up right. So right there. So that looks like a little kind of like pink layer right along top. Those are actually microvilli, okay, sticking up there. So your microvilli are sticking up, and that improves surface area, okay, for absorption of nutrients in the digestive tract. All right. So you have this structure aligns with the function of all these guys. All right, next slide. Here's some more cell specialization. You have sperm cells here, okay, and you can see the whip-like flagella, or singular would be flagellum. You can see how each of these has, and there's the head there. There's the head of the sperm. So you can see it has a flagellum sticking off, so it can wave back and forth and make the sperm take off towards the egg. Blood cells. You have white blood cells. So this is a white blood cell here. These are all red blood cells, all those guys, right? Now you notice, and I'll show you one other thing. Let's see here, these little tiny ones, the smaller ones here, there's two of them there, there's one there, one there. All those little guys, those are platelets. Okay, platelets are used for stopping you from bleeding. Red blood cells carry your oxygen. Okay, white blood cells are for immune response, so fighting off bacteria, viruses that get into you. You notice there's a ton of red blood cells because you need to carry a lot of oxygen in your bloodstream. And so that you'll notice that there's like a kind of white part in the middle. Well, the reason is, I'll talk about this later, but here's a cell. Let's assume that it's kind of round. Okay, and that there's a nucleus here in the middle, right? So you have this sphere. Well, when red blood cells mature, they eject the nucleus. Okay, so you don't have. So what happens is the cell then becomes concave and convex, and so you have this narrowed part in the middle. So when you have a cell that's laying straight down on a microscope slide and you have light going through it, and it's thicker on the edges and thinner in the middle, there's more light coming through here, so it's lighter in the middle than it is around the edges. So that's why you have like this lighter center and darker edges on the red blood cells. So it makes it easy to identify red blood cells. Okay, motor neuron. This is a nerve cell. So there's the nucleus. Cell body is the whole thing here. That's the cell body. And they have processes. These are called dendrites. Okay, and one of those processes is going to be called, one process is going to be called an axon. And let's just make it up here. It's hard to tell what it is, so let's say that this one right here is the axon. Okay, and dendrites are what a nerve cell uses to reach out and touch all the other nerve cells around it and communicate with them. An axon, it also uses that to communicate with also. But there's only one axon per nerve cell. We'll get to that later on. Don't worry about that now. I just want you to see cells have different shapes, different sizes. They do different things, and they do different things because they have different shapes. So the structure or the shape of something is closely aligned with the function of it. All right, next slide.